So we are going to discuss chapter 4, which is on enzyme, and we are going to look at part B. We are going to discuss question 7 first. Okay, so if we look at the question here, it says figure 22 shows effects of catalyst on rate of reaction. Okay, so for this question you have to label in the box which is M, which is N. So M is it shows the rate of reaction without enzyme, kan? So this one is M. As you can see, the activation energy is higher, which compared to N, which is the activation energy for the reaction that is catalyzed by enzyme. Okay, so boleh Okay, so for question number 2, it says here state 2 ways on how enzyme lowers activation energy. So this it is straight away from your notes. Okay, you have to memorize like this, right? So this is how enzyme works on lowering the activation energy. First, it brings the substrate together in the correct orientation. Okay. Second, it provides a favorable cellular microenvironment. And then third, it stretches the substrate bonds towards the transition state. And then the last one, it forms temporary covalent bond with the substrate. So there are 4 points. For 2 marks, you only need to state for 2 points only. 2 marks, 2 points. Okay. So the next question, it says here there are 2 models of enzyme action. So which of the model better at accommodating substrate? The answer will be induced feed mechanism. Okay, so you have an enzyme with the active site that is not exactly complementary with the shape of the substrate. So as the substrate binds into the active site of enzyme, it causes a slight change in the active site of the enzyme. So induced feed. Okay, induced feed mechanism is best at accommodating substrate. Okay, and then explain your answer. Why? Because the active site is flexible. Okay, the active site is flexible. It can change shape. Okay, active site is flexible. Right. Okay, so and then another point, because the binding of the substrate will induce a slight change. Okay, you don't necessarily have to include the word conformational. Okay, you can you just can mention induces a slight change. Okay, a slight change in the shape of the active site. Okay, so sekejap. Okay, so the next question, it gives you a graph which is figure 23, shows competitive and non-competitive inhibition. So basically in this graph there are 3 different reactions. Okay, so for the first graph, which is the D, the rate of reaction for reaction D, it shows you the reaction that is not inhibited by any inhibitors. Okay, so the reaction can occur normally. The rate of reaction is high and reaches maximum velocity really quickly. Okay, but for graph E, it shows you the graph with the presence of competitive inhibitors. How do you know that is the graph that shows competitive inhibition? Because as you can see, as you increase the substrate concentration, so initially in the presence of competitive inhibitors, the rate of reaction decrease from the normal reaction without the presence of any inhibitors. But as you add competitive inhibitors into the reaction, it lowers the rate of reaction. But as you increase the substrate concentration, you increase the chance of substrate binding to the active site, and therefore the rate of reaction can increase and reaches ultimately the maximum velocity of the reaction. But for graph F, it shows you for the reaction with the presence of non-competitive inhibitors. Okay, so as you can see, the more that you add a substrate into the reaction, it does not give any effects. Okay, so as you add non-competitive inhibitors into the reaction, it ultimately causes the rate of reaction to decrease and does not affected by the substrate concentration. Okay, so why? Because the non-competitive inhibitors bind to the allosteric site. It does not compete with the substrate. Okay, So the question asks which labeled graph represent competitive and non-competitive inhibitors. So I've given you the answer. So E is competitive inhibition, F is non-competitive inhibition. Okay. Okay, so next question, which label graph has the highest Km value? What is Km? Michaelis-Menten constant, which is it shows you the the rate of reaction, the substrate concentration at half Vmax. Okay, the substrate concentration, substrate is axis x kan, axis x y. Okay, so Km is Michaelis-Menten constant, which is substrate concentration at half Vmax, half maximum velocity. So yang mana? Which reaction? Is it D, is it E, or is it F? Mana? You just pull out lah kan? So the answer will be E, as the value is much more here. Okay, so you just get the max. This is the maximum velocity, kan? So this is half Vmax. So the reaction for E will have the highest Km value. Okay, will have the highest Km value. Okay. You can ask question if you want. Okay. Alright, so next question, state the advantage of having low Km value. So basically you want a reaction that has the lowest Km value. Okay, why is that? Because the smaller the Km value, it shows you the higher the affinity of the enzyme binding to the substrate. Okay, so the substrate with the lowest Km value will attract enzyme more. So it means that if you put a small amount of substrate into the reaction, the enzyme will straight away catalyze the reaction. It does not need, the reaction does not need so much substrate to be added. Just a little concentration of substrate added into the reaction, the enzyme will straight away catalyze the reaction. That is what the Km indicates. The smaller the Km value, the higher the affinity of the enzyme to bind to the substrate. Okay, so so it shows you the more strongly the enzyme binds to the substrate, and the more likely product will be produced. Okay, so basically you want a substrate with the lowest Km value, so this will attract the enzyme to bind to the substrate more. Okay. Okay, so next question, it shows you here it says explain how serine molecule inhibit acetylcholinesterase. Okay, so acetylcholinesterase is an enzyme found in between neurons at the synapse. The function of this enzyme is to break down neurotransmitter acetylcholine. Okay, so you have one neuron and then another neuron that transmit impulse. Okay, so the transmission of this impulse occurs when neurotransmitters such as acetylcholine bind to the postsynaptic neuron, the dendrite of the postsynaptic neuron, to cause impulse to be transmitted. But you do not want this transmission of impulse to occur continuously without stopped. Okay, so in order to stop the transmission of the impulse, you need to remove the neurotransmitter, which is acetylcholine, carried out by the acetylcholinesterase, the enzyme that removes the acetylcholine as the neurotransmitter that stop the transmission of impulse. So when you have an inhibitor, which is serine, that binds to the active site of the enzyme, so therefore acetylcholine cannot bind to the active site of the enzyme. Okay, so when acetylcholine cannot bind to the active site of the enzyme, the neurotransmitter is always present at the synapse and causes continuous transmission of impulse. So basically serine will function as inhibitors, but you have to identify what type of inhibitors. It is irreversible inhibitors. Okay, irreversible inhibitors, what does it mean? It binds permanently to the active site of acetylcholinesterase. It binds permanently. Irreversible maksudnya ikatan antara inhibitors dengan enzim tu kuat. Kan? Kuat. Permanent through what? Through covalent bonds. Okay, through covalent bonds. So it is a type of irreversible inhibitors. Where does it bind? To the active site. So irreversible competitive inhibitors. Compete dengan siapa? Compete dengan substrate. Substrate tu siapa? Acetylcholine. Okay. Sekejap. 2 markah lah. Kan? So the first point, it acts as irreversible competitive inhibitors that bind tightly and permanently to the active site of enzyme through strong covalent bonds. Another point, 2 point dah boleh dapat kat situ. And what is the consequences? It prevents the substrate, which is acetylcholine, from binding to the enzyme, from binding to the active site of the enzyme. Okay. So this one you have to look back into your notes. Okay, it is irreversible competitive inhibitors. Okay. So next question. Which method of enzyme immobilization is used to produce lactose-free milk? Okay, so you have different types of ways. Okay, different ways, different types of enzyme immobilization. So for producing lactose-free milk, you can use entrapment. Okay, you can use entrapment. Okay. So that's the last question for question number 7. So do you have any question for question number 7 before we proceed to question number 6? Kita start daripada soalan belakang. Ada soalan?
Yang untuk yang neurotransmitter kan. Untuk neurotransmitter lain akan jadi macam ada inhibitor yang akan inhibit dia ke?
Ada setiap inhibitors tu dia ada specific enzyme dia. So must be the inhibitors will the enzyme is affected by different inhibitors. Macam tu lah. Okay. Okay. So for this question, it is the same as the serine question here, but for this one you can see clearly the diagram. Sama je, the same question. Okay, but this one it gives you the diagram for you to be able to see clearly the interaction between serine and the enzyme. The enzyme is this one, acetylcholinesterase. The inhibitors is the serine. It binds to where? It binds to the active site. Because it binds to the active site, it is competitive. Because the interaction is through strong covalent bond and permanently bind to the active site, so it is irreversible. Okay, irreversible inhibitors. So identify the type of inhibition, irreversible competitive inhibition. Okay. Dia punya substrate dia adalah the neurotransmitter. This one here. Acetylcholine is a type of neurotransmitter that cause the neuron to be able to transmit impulse from one neuron to another neuron. But you do not want the transmission of impulse to occur continuously without stop. So to stop the transmission, you have to have this enzyme. Okay, this enzyme to remove the neurotransmitter. But if you have serine, you cannot remove the neurotransmitter. Okay. Okay, so next question discuss how serine disrupt the function of acetylcholinesterase. So tadi apa? Tadi dia punya sifat dia, jenis inhibition dia adalah irreversible competitive inhibition. So explain lah kenapa dia irreversible, kenapa dia competitive, kan, untuk 3 markah. So the first point, irreversible because why? Because the serine binds tightly, permanently, covalently to the active site of enzyme. Okay, itu point yang untuk kenapa dia irreversible lah. Tightly, permanently, covalently to the active site of the enzyme. Okay, and then so what happen when the serine binds to the active site? It prevents acetylcholine from binding to the active site. So that is the second point. It prevents acetylcholine or substrate from binding to the active site of the enzyme. Okay. So what is the consequences? Acetylcholine cannot be broken down. Okay, the neurotransmitter cannot be broken down or cannot be removed from the synapse. Okay, or it causes accumulation of acetylcholine in the synaptic cleft. So what is the consequences of this accumulation of the neurotransmitter? It causes continuous impulse transmission. Okay, it causes continuous impulse transmission. Kenapa awak tak nak continuous impulse transmission? Bayangkan kalau impulse transmission tu continuous and causes continuous contraction of muscle. You do not want continuous contraction of muscle. Awak nak duduk tiba-tiba dia jadi kejang kat situ, faham tak? So you do not want continuous transmission of impulse. So that's why you have to remove the neurotransmitter. But when you have serine, you cannot remove that neurotransmitter. Okay. So those are the answers that you can include in your answer. Okay, those are the points. Okay. Okay, so next question. It says here feedback inhibition. Okay, feedback inhibition is one of the way to regulate enzymatic activity in cell. Okay. Okay. So let's continue to discuss. Okay, question B, feedback inhibition is one of the way to regulate enzymatic activity in cells. Okay, so figure 20 shows the simplified metabolic pathway of CTP. Okay, so CTP is similar to ATP lah. It differs in the base that binds to the molecule. Okay, CTP is cytidine. Okay, so ADP is adenosine. Okay, both molecule has 3-phosphate bind to it. So it is a high energy molecule. Alright. So the keyword that you want to look here is feedback inhibition. Okay. So you have to know for the concept of feedback inhibition, you have a series of reaction occurs that involves many enzymes in the reaction and many formation of intermediate products until you form the end products. The end products is the CTP molecule. Okay. So through the concept of feedback inhibition, you know that the end product will become the inhibitor of the first enzyme of the reaction. So the purpose is to stop wastage of chemical resources in cell. So bila cell tu dah hasilkan enough certain molecule at certain time, so it will stop production of that molecule for a while to let the cell to use that molecule. Okay, so the question it says here, CTP can act as allosteric inhibitors. Okay, allosteric inhibitors, it binds to the allosteric site of the first enzyme of the reaction because it is feedback inhibition. So determine which enzyme the CTP molecule acts on. The first enzyme lah, the first enzyme of the pathway, which is enzyme 1. Okay, enzyme 1. Alright, so the end product will bind to the first enzyme of the pathway. Okay. So next question, CTP concentration may be high or low, okay, depending on the cell's demand. Keperluan sel tu lah. Alright. So explain how the changes in CTP concentration could affect the rate of N-carbamyl aspartate. Okay, so N-carbamyl aspartate is the intermediate product produced in the chain of that reaction. Okay. So this is the intermediate product. Okay, intermediate product, this is the final product. So this is the initial substrate. Aspartate is the initial substrate. But the question asks you for the intermediate product production. Okay, the N-carbamyl aspartate. So you can choose which one to answer. You can answer if the concentration of CTP high or if the concentration of CTP low. If you choose to answer CTP high, the concentration of CTP is high, so you can explain on the way of how CTP becomes the allosteric inhibitors for the first enzyme. Okay, so you can explain one for the first point. When concentration of CTP is increased or high, it causes what? CTP to bind to the first enzyme of the reaction, okay, inhibiting the enzyme 1. It inhibits enzyme 1. Inhibit enzyme 1. Okay, it inhibit enzyme 1. So what is the consequences? The concentration of N-carbamyl decrease. Okay, the concentration of N-carbamyl decrease. N-carbamyl decrease. So this is for the first point lah. Satu markah kat sini. So but if you choose to explain if the concentration of CTP is reduced, so when CTP is reduced, so the cells needs to produce more CTP. So so the first point that you can say that CTP concentration is low, kan? So this activate the first enzyme. Okay, it activates the first enzyme. CTP activates enzyme 1. Okay, enzyme 1. So what is the consequences to the concentration of N-carbamyl aspartate? Increase. Faham? Okay. So basically you have to know for feedback inhibition, the end products function as the inhibitors for the first enzyme if the concentration of end product increase. So for this question, you can mention either one. If increase, what happened? If decrease, what happened to the intermediate product here? Okay. Okay, so next question. Lactase breaks down lactose into simple sugar and often immobilized for use in the food industry. A student carried out an investigation on production of lactose-free milk by immobilizing lactase in small arginate beads as shown in figure 21. Okay, so you have a container here and you fill up the container with alginate beads that you bind the alginate beads with lactase. Okay, lactase. So and then you pour milk through this container that has alginate beads with lactase attached to it. Okay, so what do you want as the product? You want to produce milk that is lactose free. Okay, lactose free milk. So what is the function of this lactase? It breaks down the lactose into its monomer, which is what? Galactose and glucose. Okay, lactose and glucose. So this lactose free milk will be much more sweeter than the normal milk. Okay. So that's answer for the first question. Name 2 monosaccharide present in the lactose-free milk: galactose and glucose. Okay, so second, describe the advantages. So you have advantages for 2 marks. So means that you have to give 2 advantages. Okay, so look at the keywords lah, kan? So 2 marks, 2 advantages. Okay. So what are the advantages of using immobilized enzyme? So you can reuse the enzyme over and over again for many reactions. Okay, so enzyme can be used repeatedly for many times. Okay, repetitive usage of enzyme. That is the first advantage. Okay, for the second advantage, because you can use the enzyme over and over again, so therefore it is more cost effective. Okay, cost effective. You can save the cost of producing more enzyme. Okay, so that is the second point. It is more cost effective. And then another point that you can include, basically the purpose of using immobilized enzyme so that you can easily separate the enzyme from the product. That is the purpose. Okay, so the product is not contaminated or mixed up with the enzyme. So that is another point. Enzyme does not contaminate the product, which is the lactose-free milk. And then another point that you can include in your answer, for immobilized enzyme, the rate of reaction is higher. Okay, the rate of reaction is higher. It produces more product. Another enzyme is more stable. Okay, enzyme is more stable, and then it speeds up the reaction. Okay, speeds up the production process. Okay, so those are the answers that you can include in your answers. Those are the points. Okay. Okay, so next question suggests why student choose small beads okay, rather than large beads. Okay, imagine kalau this is the small bead that is used during the experiment. Imagine if you use the bigger one, bigger beads. Okay, so bigger beads is less effective. You want the smaller beads to be used in the reaction. Why? Because by using smaller beads for the first point, the smaller beads provides larger surface area. Okay, larger surface area. You can attach more enzyme. You can attach more enzyme, right, to the beads. So when you attach more enzyme, because it provides larger surface area, so therefore rate of reaction is higher. Then second point, rate of reaction is higher. Okay, so you can pack more beads. You can that is the point. You can pack more beads. More enzyme is attached to the beads, and then if you use more beads, the milk that passes through these beads will be slower. And so as you pour the milk through the beads, the milk will pass through the beads much more slower. Okay, so kenapa nak kena aliran susu tu kena lebih perlahan lagi efektif? Because this increases more time for the reaction to take place, kan? So more time for enzyme to be exposed to the milk or to the substrate. Okay, so lagi banyak masa milk tu boleh react dengan enzyme, so that to make sure more reaction can take place. Okay. Okay, so that's the end of question 6. Okay, so any questions so far for question 6?
No.
Ada. Okay, so let's look at question 5. Okay, question 5. Kita buat pada belakang lah ya. Okay, so figure 17. Okay, for question A, figure 17 shows the induced feed model of an enzyme. Identify K and L. So easy. This one, K is what? K is the substrate. L is the substrate enzyme complex. So this is substrate enzyme complex. Substrate binds to the active site. So you have substrate enzyme complex. Okay. So that is the first question. For the second question, describe the mechanism of this enzymatic reaction. So as you can see, if you can see in this question is actually, if you can see in the diagram, the active site is not exactly complementary with the shape of the substrate. As you can see there, macam ada pada pandangan maybe dia dah mention dah awal-awal kan soalan tu, sorry. Okay, dia dah mention initially in the question it mentioned in the question it is induced feed model, kan, induced feed model. So for this question you have to explain on the induced feed model lah. So the first key point that you need to include in your answer, as the substrate binds to the active site of the enzyme, it causes a slight change. Okay, a slight change in the shape of the active site. That is number 1. Okay, so what is the significance of this slight change? It brings the enzyme in the correct orientation with the substrate. Okay, it brings the enzyme in the correct orientation with the substrate to form enzyme substrate complex. Okay, and leads to the production of the products. So once product has been produced, okay, so the product will be released from the active site, and the active site returns back to its original shape or uninduced shape. Okay, so as the product is released, the enzyme returns back to its original shape. Okay, so that is the answer. Okay, question number 3. Suggest how low pH could affect the structure and function of enzyme. Low pH, what condition is this?
Acidic.
Acidic. So acidic condition is due to what concentration that increase or lowered?
H plus concentration.
H plus concentration increase. Okay, so low pH condition indicates that there is a high concentration of hydrogen ion. Okay, so first point. Okay, first point, low pH increases high concentration of hydrogen ion. First point. So when there's a high concentration of hydrogen ion, it lowers the rate of reaction. Second point. Okay, it lowers or decreases the rate of reaction. Kenapa? Why? Because the presence of this high concentration of hydrogen ion will alters the ionic charges, the ionic charges on the active site of the enzyme, and causes the enzyme to be denatured or dysfunction. Okay, it can cause the enzyme to denature or dysfunction due to the breaking of hydrogen bonds or ionic bonds in the active site of the enzyme. Okay, so that is the consequences. Okay, so high concentration lowers the rate of activation because it alters the ionic charges on the active site and breaks the ionic bonds in the active site of the enzyme, causes the enzyme to be denatured. Okay, so next question. The equation below shows the breakdown of dihydrofolic acid to folic acid. So dihydrofolic acid is the substrate, folic acid is the product. Okay, dihydrofolate reductase is the enzyme. Okay, so state the class of enzyme that catalyzes the above reaction. So basically we know that there are 6 classes of enzyme. So from the name of the enzyme, you can identify straight away the class of enzyme, reductase. So it means that the enzyme catalyzes redox reaction. So it is oxidoreductase enzyme. Okay, so the class will be oxidoreductase enzyme. Second question, why dihydrofolate reductase only binds to folic acid? So this is basic concept. Every enzyme is specific to catalyze specific reaction. So that's the answer lah. Enzyme are substrate specific. Okay, every enzyme binds to specific substrate due to the specific active site lah, the specific shape of the active site that can only bind to specific substrate. Okay, enzyme catalyzes specific reaction. Okay. Okay, so next question. The antibiotic trimethoprim has structural similarity to the dihydrofolic acids. Okay, so we know that this one is the inhibitor. Okay, inhibitor keyword, another keyword that you have to look out for is similar. So it is competitive inhibitors. Okay, competitive inhibitors. So explain how the trimethoprim inhibit the enzyme. So the keyword we have identified already, it is competitive inhibitors. So just explain lah from this word kan. So the inhibitors or the antibiotic binds to the active site of the enzyme. Okay, it binds to the active site of the enzyme. Second point, kan first point yang ni competitive. Second point, binds to the active site. Active site of the enzyme. And then another one, apa consequences dia? It prevents the substrate, which is the defolic acids, to bind to the active site of the enzyme. Prevents the dihydrofolic acid or the substrate to bind to the active site of enzyme. Okay, ada 3 point lah kat situ. Another point that you could add, the antibiotic trimethoprim will compete Okay, it competes with the substrate to bind to the active site. Compete. Another point, you have to explain on the competition part. Compete. Compete to bind to the active site. Okay. So ada 4. There are 4 points that you can write there for 3 marks. Okay, so next question. Figure 18 shows how enzyme activity changes with temperature. So for this question, you just look at the diagram lah, kan? Describe the relationship between the effects of temperature and the reaction. So as the temperature increase, the rate of reaction increase. Okay, so that is the obvious point that you can write. As temperature increases, the rate of reaction increases up to 40 degrees only. Okay, up to 40 degrees only. Ataupun the optimum rate, the optimum temperature for the reaction will be 40 degrees Celsius. Okay, optimum temperature. So another point that you can write, or you can also write above 40 degrees, the rate of reaction starts to decrease. Okay, so above 40 degrees Celsius, the rate of reaction starts to decrease. Or above 55, there is no rate of reaction. Okay. Okay. Tadi dia stuck. Kita habiskan soalan ni sikit. Okay, so next question, explain what happens to the enzyme above 40 degrees Celsius. Above 40, as you can see, above 40 degrees, the rate of reaction starts to decrease. Kenapa? When you increase the temperature, you increase the collision, okay, between the molecules in the enzyme. So this causes hydrogen bonds or the chemical bonds within the enzyme to be broken. Okay. So this leads to the enzyme to become denatured, second point, and changes the shape of the active site of the enzyme, and therefore substrate cannot bind to the enzyme. Okay. So this one you explain on the denaturing part of the enzyme. Okay. So straightforward question. So far, do you have any question? Tak ada? So I guess we can stop to this question number 4, or do you want to continue? Sebab lepas ni saya takkan discuss dah. So saya nak habiskan on the 2 chapters. 4, kita habiskan sikit. Question number 4 here. Figure 13 shows a reaction in 2 different conditions. Circle A or B to indicate the reaction with enzyme. So obvious, yang ni jawapan dia senang je kan. So B is the reaction with enzyme. Okay, because the EA, the activation energy, is lower. Number 2, enzyme concentration and substrate concentration are factors that affects the rate of enzyme reaction. So what happened to the rate of reaction if substrate is the limiting factor? So you have limited amount of substrate, so therefore there is no change in the rate of reaction. Okay, no change in the rate of reaction. Number 2, name 2 classes of enzyme according to the Nomenclature Committee of International Union of Biochemistry and Molecular Biology. So ada 6 kan, so you can name any N2. Okay, so we have ligase, oxidoreductase, transferase, hydrolase, lysase, isomerase. So 1, 2, 3, 4, 5, 6, ada 6 kan. So nak 2 je. Okay. And then next question, ferrum and zinc ion are cofactors. So why do apoenzyme needs a cofactor? So basically you have to know the definition of apoenzyme. Apoenzyme is inactive enzyme without cofactor binding to the enzyme. That is apoenzyme. Kalau ada cofactor, then we call it as holoenzyme. Okay. So why does APO enzyme needs a cofactor? Cofactor function to aid or assist in the enzyme reaction. Okay, it assists. Okay, it assists the enzyme to be activated. Macam itulah kan. So enzyme is in active form of enzyme. So it requires cofactor to activate the enzyme, to assist the enzyme reaction. Okay. So next question, differentiate reversible and irreversible inhibitors. So for 2 marks, so you have to differentiate and give 2 differences, reversible and irreversible. So for the first point, kenapa inhibitors tu irreversible? Kena cerita dia dari segi interaction inhibitors dengan enzyme. Kalau kalau reversible, through weak bonds, weak interaction. Okay, the interaction between inhibitors and the enzyme is through weak bonds. Tapi, such as apa weak bonds? Hydrogen bonds. Okay. Tapi kalau irreversible, the interaction between the enzyme and the inhibitor is through strong bonds, strong covalent bonds. Okay. So that is for the first point. Second point, macam mana inhibitors tu bind pada enzyme tu? Kalau weak bonds, obviously the binding will be loose binding. Okay? So the inhibitors bind loosely to the enzyme, kan, bind loosely. Tapi kalau yang irreversible, bind tightly, permanently to the enzyme. Okay. So another point that you can include in your answer. Sorry. Okay. For reversible, enzyme can revert back to its functional structure. Kalau reversible inhibitors tu tercabut ataupun terkeluar, enzyme can function again. Kan? So enzyme can revert back to its functional shape, functional state. Okay, functional structure. But for irreversible inhibitors, enzyme is totally disrupted. Kan? So enzyme unable to function anymore. Okay, cannot function anymore. Okay, so next question, identify the type of allosteric regulation shown in figure 14. So kat sini dia adalah sejenis apa? Cooperativity. Kan? Cooperativity. What is cooperativity? You have allosteric enzyme having several subunits with several active site. So this allosteric enzyme, dia dalam keadaan tak stabil. It keep on changing from the inactive form to active form. Inactive form to active form. So you have to stabilize the enzyme to make sure that the enzyme is in the active form. Okay. So in order to activate the enzyme, you need only one substrate to bind to one of the active site. So that is cooperativity. Okay, cooperativity. Cooperativity, substrate binds to the active site of enzyme to activate all subunit of the enzyme. So that is cooperativity. Okay. Kalau kita tengok dalam nota awak, you have to know on cooperativity satu. Satu lagi, activation of the enzyme or inhibition of the enzyme in the presence of allosteric inhibitors. Okay, kita panggil allosteric regulators. Okay, allosteric regulators. Allosteric regulators ni ada 2, inhibitor, lagi satu activator. Yang ini 2 ni dia bind kepada allosteric site. Okay, allosteric site. Beza tau. Beza dengan cooperativity. Cooperativity substrate. Okay, cooperativity melibatkan substrate. Tapi allosteric regulation melibatkan allosteric regulators binding to the allosteric site. Okay, fixing either in the inactive form of enzyme or the active form of enzyme. Okay. Okay, so next question. Figure 15 shows line Weaverberg plot of an enzyme reaction. Determine the value of Vmax. Kan? So value of Vmax, line Weaverberg plot ni adalah double reciprocal kan. So you have to find the value kan, y-axis ni. So Vmax kan, so 1 per berapa value dia? 0.02. Jawapan dia berapa? 50 kan, 50. 50, in this case dia tak bagi unit, tapi kalau dalam soalan dia bagi unit, you have to write the units. Okay, you have to write the unit for the subs, for the rate of reaction. Okay, for the rate of reaction. Okay, sama juga kalau soalan dia suruh cari value Vmax, bagi unit, km pun nak kena bagi unit juga. Okay. So, okay. The next question, figure 16 shows a method of enzyme immobilization. So, describe the method. Okay. So, this method, kalau tengok dia apa? Dia adalah jenis apa ni? Entrapment. Okay. So, enzyme is trapped in a 3D lattice. First point. Okay. Because the enzyme is trapped, so enzyme is not chemically bound to the to the container tu lah kan. So enzyme is not chemically bound. Okay, so polymer includes alginate and also silica gel. So describe sikit lah pasal entrapment tu. Okay, so habis buat question 4. So far is there any more question that you need to discuss? Ada tak soalan untuk yang 1, 2, 3? Ada tak yang you all nak saya discuss specifically? Sebab yang lain tu saya rasa dia repetition je konsep dia. Penicillin ni okey tak? Penicillin ni saya dah discuss ke belum? Belum kita tengok ni sikit je. Penicillin is okey. Ini kan soalan ni panjang sangat. Dia sebenarnya nak jadi distraction je. Yang awak kena take note, ini kan ayat ni semua distraction. 6, ini distraction kan. So apa yang you all kena tahu, penicillin acylase is an enzyme used to prepare 6-APA, okay, from naturally occurring penicillin. So in order to produce large quantity of 6-APA, penicillin acylase, which is the enzyme, is immobilized. Okay, so table 1 shows the rate of reaction when using penicillin acylase in solution when immobilized at different temperature. So dia bagi dua condition. Satu, dalam keadaan enzyme tu dalam solution, not bound to certain surface. Okay, so yang second reaction ni, penicillin is immobilized. It is bound to certain surface. Alright. So ada temperature dia manipulate from 20 to 70. So for the enzyme that is not bound, as you can see, as you increase the rate of as you increase the temperature, you observe that the rate of reaction mula dia increase kat sini, tapi decrease as the temperature exceed 50 kan, exceed 50, they start to decrease. For the immobilized reaction kan, so dia akan increase, tapi bila above 60, they start to decrease. So yang ini sampai ini increase, rate of reaction increase. But bila 60 sampai ke 70, rate of reaction start to decrease. Nampak tak? Boleh nampak ke? Mula temperature 20 increase, 30 increase, 40 increase, 50 increase, 60 increase. Kalau tengok immobilized enzyme, dia lagi stable as you increase the temperature. Dia lagi stable. Kalau 60, the temperature increase lagi. Tapi kalau not immobilized, bila exceed je 50, dia start to denature. Kan? So maksudnya kalau immobilized enzyme, it is more stable lah. It is more stable. It can withstand a higher temperature compared to the normal reaction. Okay. So based on table 1, explain why the rate of reaction of penicillin acylase in solution decreases, okay, between 50 and 60. Between 50 and 60 in solution. Maksudnya not immobilized kan? It is not immobilized. Okay. So kenapa? Sebab due to the chemical bond within the enzyme that is broken. Okay, the chemical bond that holds the enzyme structure is broken and causes the enzyme to be denature. Itu je lah kan. Enzyme denatured because because the bond within the enzyme is broken, or the active site of enzyme is altered. Is altered. Okay, so those are the point. Above 50 je due to high collision kan, so it causes the chemical bond to be broken, enzyme to be denatured. Okay, next question. Describe the rate of enzyme activity at different temperature for immobilized penicillin enzyme. So yang ini awak kena tengoklah tadi kan. So ada dua keadaan. Satu daripada temperature 20 dan 20 sampai ke 60 degree Celsius. The rate of reaction increase ke decrease? Rate of reaction?
Increase.
So 60 to 70, what happened to the rate of reaction?
Increase.
Dua markah. Satu markah 2 hingga 60 degrees increase, 60 to 70 decrease. Okay, one more each. Okay, next question. One method of enzyme immobilization is adsorption. Describe how enzyme is immobilized using this technique. Immobilization through adsorption. So this one involve apa? Formation of weak bonds such as hydrogen bonds, van der Waals interactions, hydrophobic interaction between the enzyme and the support. Okay, and the support. So 1 mark lah kan. So it involve the formation of weak bonds such as hydrogen bond between the enzyme and the inert support, or the resin ataupun silica gel alginate. Okay, so that is adsorption. It involves formation of weak bonds between enzyme and the support. Okay, next question. Number 4, explain another advantage of using immobilized enzyme. Sama je macam point tadi. Kan, advantage of using immobilized enzyme, you can use the enzyme over and over again. It is more cost effective, and then you can separate easily the enzyme from the products. The enzyme does not contaminate the products. The enzyme is more stable. Macam tadi kan, as you increase the temperature, the enzyme still does not denature. Okay. So itulah dia. So those are the point that you can include as the advantage of using immobilized enzyme kan. So I think that's it. Ada soalan lagi? Yang lain ni semua sama je lah kan. Characteristics of non-competitive inhibitors tu kita dah go through kan.